Thanks again, Andy. So James and I have only 10 minutes for the wrap-up, but I think that's good for you because it's already been a dense, concentrated day, hasn't it? I'm going to show just a few slides to summarize some of the new directions that we see as being tenable in GIST. But before I do, I can't resist to follow up on Andy's comments. I think it's really a bad thing that societally we have made academia out to be the shining knight, and industry, on the other hand, pharma, is kind of an evil for-profit enterprise, as though that would be such a terrible thing in a capitalist society. But the truth is that we complement each other beautifully, and you absolutely need both. In industry, as one example, I think one is unlikely to find a group that for a sustained period of time, like 20 years, 30 years, is going to obsess about liposarcoma, about GIST, and not just do something one day and then drop it the next day when orders come down from on above. And in academia, very few of us are going to be synthesizing large libraries of compounds and figuring out which does which and the properties, the pharmacological aspects of the drugs. If you want to go forward, you absolutely need both camps for the reasons that Andy discussed and for more. So I think we have to do, the only reason I said it is that there's a lot of red tape that gets between us working with industry, us being the academic folks, that I think is unnecessary, and it's driven by a well-intentioned notion that industry is opportunistic. Well-intended, we could talk about another time, but I think this needs to be rolled back a bit, but my 2 cents. So future directions for GIST, we've discussed the fact, of course, that the drivers in GISTs from the very get-go are the activated forms of KIT and PDGFRA, these so-called kinases that send their signals into the cell. And it's only common sense that since they are the drivers, they're the engines, that we continue to focus upon those as being the low-hanging fruit. And I'm just going to show again, a testimonial to how much the patients have done to drive this forward locally. This is, each of these boxes is one metastasis from a patient who had a surgery, a GIST patient, of lots of different metastases all at once. And each of those codes is telling us a different resistance mutation. We found one resistance mutation in multiple metastases that we already knew about in GIST, and we found 6 novel ones that we'd never encountered before. So imagine what that means if you find new resistance mutations you weren't even aware of, and now you have a group of drugs, and you want to know how well do they shut down resistance mutations. The more representatives you have, then the better a job you can do of defining which is the winner, which is the most effective drug. So here, if we take this patient's, each of those dots is a metastasis from that patient, and the blue boxes indicate different resistance mutations, different second mutations. And here we're asking, what drugs would shut down all of them if you wanted to put this patient with those resistant mutations into remission, stable disease for a long period of time? Green means it's sensitive. This isn't working. Yellow means a kind of intermediate response to drug, and red means it's just flat out resistant. Well, we weren't surprised as the patient who progressed after imatinib and sunitinib that those drugs didn't work very well. Here's sorafenib, which in this patient wasn't much better. And here are 2 drugs that, at least on paper, based on our lab testing, look fantastic because they shut down every single one of those resistance mutations. DP3636, I'll come back to it in the summary slide, is an example of a drug that inhibits KIT directly, but binds to a different part of the protein from the classic drugs, imatinib, Gleevec, Sutent, and so forth. It's a novel way of getting at KIT. And STA9090 you already heard about. Here we have a dissociation from clinical reality because this is one of the HSP90 inhibitors that has been frustratingly promising in the laboratory and hasn't been brilliantly successful in the clinic. And what it's told us is HSP90, great thing to target, but we need to learn how to do it the right way, in a more effective, persistent, selective way without the same level of toxicity, and keep HSP90 down, because every one of these resistance mutations needs HSP90. If you shut down HSP90 the right way, it will certainly work. Okay. And then Thesar, in his talk, highlighted the fact that the so-called MAP kinase pathway has now been validated as being very important in causing GIST cells to grow. The only reason I mention that, 2 reasons really, is that in the past much of our attention has been on a different pathway downstream of KIT. PI3 kinase goes to AKT, also very important in enabling GIST cells to grow and to survive, and there's some clinical trials that some of the speakers addressed that are beginning to ferret out how well will those inhibitors work in GIST. But the MEK-MAP kinase hasn't received so much attention. There are inhibitors. They haven't been fantastically successful in clinical trials against most other cancer types in people. What's different about GIST? Why are we excited about this? Because unnoticed until about a year or two ago, this pathway is absolutely essential in determining what makes a GIST, creating the GISTness of a cell. If you block the pathway, the cells no longer grow well, as Thesar said, and they're really no longer a GIST. So it's a crucial pathway. What's the problem with this one? Because it's so important, the cells have a number of elegant checks and balances, feedback loops we call them. So if you target the pathway, the cells are able to step sideways and reactivate it by one of their feedback loops, okay? Segue to Adrian, who gave his talk about how you classify GIST, but the thing he's really doing brilliantly that will change the world is asking for 20,000 different genes in GIST all at the same time, which are the ones that matter. Adrian's taking the GIST cells where we target that pathway and then asking, what's the second thing that you can hit that will block the feedback loops, that will convert this to something like the home run that James was mentioning? Okay, and then the question came up about antibodies, so I couldn't resist putting these slides in, okay? Here's a breast cancer cell, the one with blue, amongst some, there I've got it to work, against some normal cells. And the extra brown spots are extra copies of the HER2 gene, another kinase gene, which is often amplified or activated in breast cancer. And the blue here, this is a bad system. I'm going to switch. Okay, the blue, good thing it's the end of the day. There we go, bravo, just about. I'm bringing my own pointer next time, okay? See this dark circle? That's the protein product of the gene which is activated, the HER2 protein, and it's at the cell surface where we expect it to be. KIT, whenever we show it, is supposed to be at the cell surface, and the slide I wanted to show you is, here's the reality, okay? 3 different metastases from that patient who had the 46 metastases that I showed a few slides ago. And they were in groups of patterns. So the brown staining is showing us, I'm going to do it the old-fashioned way, with the finger. The brown staining is the KIT protein in the cells. But what you see here is that none of them have that ring around the exterior of the cell that would tell us that the KIT protein is present at the surface with part of it sticking out. Almost all of the KIT protein in GIST cells is stuck inside the cell. And then the question becomes, if you have an antibody therapy which can only attack from outside the cell, the antibodies don't get inside the cell, will that inhibit enough of the KIT to make a difference? I'm always optimistic, so I don't want to say because of this that it isn't going to work, but we'd be foolish not to recognize that it's a little bit more complicated than the HER2 example in breast cancer, okay? So as future therapies that we think could be broadly effective, even when we keep in mind the heterogeneity of resistance mutations, we have more selective ways of targeting HSP90. We're working on that, and one has to make progress with that because it's a great approach when it works. The new approaches to drugs that bind with KIT directly, the drug I mentioned that I hope will go into clinical trials this coming year, they're called switch region inhibitors. Again, they interface with a different part of the KIT protein. And then the downstream targets that are relevant, no matter what the resistance mutation is, you still have to signal through PI3 kinase. You still need that MEK-MAP kinase pathway. So those are great places to target, but it isn't just like take a sledgehammer and hit the pathway. You have to figure out how to do it in a way which is selective for GIST. One of the nuances of the pathway that matters a lot in GIST doesn't matter a lot in normal cells in the body, because we also have to be realistic and say, when we find the next Gleevec that works, it may also be something that people need to be able to stay on with acceptable toxicity for 5 years, 10 years. It can't just be a matter that you can put up with it for a couple of months. So we need a very precise, selective way of targeting the pathways. And there are a few more that, for lack of time, I'm not going to go into, but I just want to show them, so I hope you're encouraged that the machines that break down these cells have shown us vulnerabilities that we can now convert with hard work into other drugs that complement Gleevec. The immunotherapies I spoke about. And then, once again, we can take a few questions just to close, but we can't thank you enough, all of you who support the clinical trials and the work that's done on the material, whether it's having the surgery and saying, yes, people are free and encouraged to do all the things that are important to do with this material to show the way to the future. And for those of you, an extra special thanks who support the Just Cancer Research Fund, who in turn do so much to fuel these research efforts. Thank you for all of the above.